Hello all. So today I will be teaching one of the five kingdoms proposed by R.H. Whittaker, that is Kingdom Fungi. So biological classification চ্যাপ্টারটা থেকে এই ফাঙ্গি কিংডমটা নিয়ে অনেকেরই প্রবলেম আছে হয়, তো এটা একবার ডিসকাস করে নেবো তাহলে। এটা একটা ওয়ান শর্ট কাইন্ড অফ a video. So we know that fungi is eukaryotic and it is mostly multicellular. Why I am saying it is mostly multicellular? Because there is an exception, that is yeast, which is unicellular, right? Okay. So fungi are saprophytic as they derive nutrition from dead and decaying organic matter. So we all know that fungi produces spores under unfavorable conditions. So what is a spore? To understand that, first we have to understand what is a hyphae. Hyphae, right? Hyphae. What hyphae are? Filamentous structures like this, right? And when hyphae forms network, it is called mycelium. It is somewhat like this. Mycelium grows horizontally, but when the mycelium starts growing aerially, we call that fruiting body. So over the fruiting body, there is this covering called sporangium, inside of which the spores develop. So spores, there can be different types of spore. If it can be exogenous, if there is outside the spore, if there is no covering. It can be endogenous if the spore generates inside a covering. Again, spore can be motile and non-motile depending if it contains a flagella or not. If it contains a flagella, we call it a zoospore, and if it do not contains a flagella, it cannot move. We call that aplanospore. So revising that once again, spores are always haploid, that is N, right? If it contains a flagella, if the flagella is present, if the spore can move, we call that zoospore. Anything when we there is a zoo double o zoo term present, it means that it can move, related to movement, motility. If a flagella is absent, there is the spore cannot move, and we call that aplanospore. If there is a covering present outside the spore, we call that endogenous spore. If there is no covering outside the spore, we call that exogenous spore. Now this is important. Spore can be of two types: sexual spore and asexual spore. Right. If the fruiting body is diploid, now we know that spores are always haploid. If the fruiting body is diploid and spores forming from it, then by now we know that if the fruiting body is diploid, it must undergo what kind of cell division? Meiosis, right? To form haploid spores. So now if the spore is produced by meiosis, we call that spore sexual spore. And if the fruiting body is haploid, then the spore must have been produced by what kind of cell division? Mitosis. Now, if the spore is formed by mitosis, we call that asexual spore. These are fundamental things, and we need to know that before going into the depth of the chapter. Now, hypha can be of those filamentous structures, like hypha are these filamentous structures. Now, if the hypha can be of two types. If the hypha if there are walls present, we call that septate hyphae. And if there is no walls, there we call that aseptate hyphae. Now, aseptate hyphae, how is it formed? The karyogamy has happened, but there is no cytokinesis. Yeah, no cytokinesis. That's why it is continuous, sinusotic, or multinucleated. Now, this kingdom fungi, it contains under the kingdom fungi there comes four phyla. We can remember it by this mnemonic called PABD, PABD, right? P stands for Phycomycetes, starting from the most simplest one. Phycomycetes is the simplest one. Then there comes Ascomycetes. Then there comes Basidio, and lastly Deuteromycetes. So example of accepted fungi is Phycomycetes, and while the examples of septate fungi are Asco, Basidio, and Deuteromycetes. Now, what is the life cycle of sexual reproduction in fungi? Suppose there are two myceliums, one is male and another is female. Both the mycelium produces gamete. This is the symbol of male gamete, this is the symbol of female gamete. After the fusion of both these gametes, okay, so the first step of sexual reproduction is plasmogamy. So as the name suggests, gamy means fusion. And fusion of what? protoplasm, plasma, that is the fluid. So the first step is plasmogamy. Now the product of plasmogamy is dikaryon. What is a dikaryon? Karyon means nucleus and di means two. So plasmogamy means the fusion of protoplasm only. But see, the nucleus is clearly they are not fused till now. So there are the two nucleus, one coming from the male gamete, another coming from the female gamete. The product is dikaryon. Ploidy is n plus n. Please note that it is not 2n, because 2n zygote will be formed after the second step, that is karyogamy. After the two nucleus, there is only one nucleus. The nucleus, two nucleus have fused. This is 2n zygote. Now the third step is meiosis inside the zygote. So inside the zygote, meiosis will happen, and after meiosis, spores will be formed, which are haploid. Now this haploid spores will again germinate to form, to give rise to mycelium. So this is the life cycle of sexual reproduction in fungi. Now, before the beginning of every chapter, we must remember the examples. So under kingdom fungi, there comes four phyla, which are those starting from the most previous one, that is Phycomycetes, Asco, Basidio, Deutero. You can remember it by this mnemonic PABD, PABD. Okay. Now we will remember the examples by some trick. Phycomycetes, just remember that it is the most previous fungi, and the most previous human, who was the most previous human on this earth, he was Ram. Like, this is just a trick, just remember it. Ram, right? So R for rhizopus, A for albigo, M for mucor. Then there comes ascomycetes. Asco means sack. So just imagine there is a boy going to a picnic carrying a sack in his hand, and inside the sack, as he is going to a picnic, there must be things for refreshment. So the things are yeast, because from yeast we know we can get bread, and we can also get ethanol, this kind of things. And then there are morels and truffles, which are edible fungi, right? In that sack we have morels and truffles. Now see, after ingesting all these things, we might suffer from some kind of disease. So for antibiotic, there is penicillin. We know penicillin comes from a fungi called penicillium, that also comes under ascomycetes. Then the boy who was going for the picnic will start sitting for asan, ACN, right? A stands for aspergillus, C stands for claviceps, right? Now remember, claviceps has got some hallucinogenic property. And lastly, N stands for neurospora. Important because neurospora is generally used in biochemical and genetic research. The third phyla that comes under the kingdom fungi is Basidiomycetes. Just remember this trick, or you can remember the examples as it is. आगरा यूपी में है पर ताज से दूर. आगरा से आएगा agaricus, U ustilago, P for Puccinia, M for there's nothing from M, P puffball, and T last T उससे आएगा toadstool. So agaricus, ustilago, Puccinia, puffball, and toadstool. Lastly, there is this phyla Deuteromycetes. Just remember this trick, cat. C for Coletotrichum, A for Alternaria, and T for Trichoderma. Practice these examples for quite of times, and then you will get a grip of it. So starting with the most initial phyla, that is Phycomycetes, right? We by now we should remember the examples. We know this trick rum. So R say rhizopus, A for albigo, M for mucor. From mucor we can get the disease called black fungus, which was very prevalent during the ending of COVID. So it is also called black fungi. Albigo is a parasitic fungi on mustard leaf, and rhizopus is a bread mold. So what kind of hypha we are getting from Phycomycetes? The hypha is accepted. See, there is no walls between the cells, so it is accepted, continuous hypha. The spores are of, it can be of two types: zoospore containing flagella, aplanospore containing no flagella. Spores, uh, depending on the type of fruiting body, uh, if the fruiting body is haploid, it is producing spore which is asexual, because here the spores are produced by mitosis, and the asexual spores are endogenous. That means there is the asexual spores are under a cover. If the fruiting body is diploid, then it must produce the spores by meiosis, and then the spores become sexual, and those spores are also again under a cover, and thus we are both the sexual and the asexual spores are endogenous. The most important point that we should remember under this Phycomycetes phyla is that the three steps of sexual reproduction that we studied in the last slide, that was first was plasmogamy. uh, the product of plasmogamy was dikaryon. The second step was karyogamy, and the third step was this meiosis in the zygote. The point which is for the most important in under Phycomycetes is that in Phycomycetes, plasmogamy is immediately followed by karyogamy. Plasmogamy and karyogamy happen side by side very spontaneously, and that's why we cannot see the dikaryon in Phycomycetes. If we zoom inside the life cycle of Phycomycetes, just suppose there are two mycelium, both of them must be aseptate, because we know Phycomycetes is aseptate. So both the myceliums are aseptate. They are producing a male gamete and the female gamete. They are fusing. See, the first step is, remember the first step is plasmogamy, right? Plasmogamy is happening. But the moment plasmogamy is happening, at the same moment, karyogamy is also happening. So you see there is no dikaryon visible. This is a very, very important point for Phycomycetes. Dikaryon is not visible in Phycomycetes. So there is no N+N dikaryon. We are directly getting a diploid zygote, and that diploid zygote in case of Phycomycetes, it is called zygospore. You can see outside the zygospore a thick covering is there, and inside the zygospore meiosis is happening to form spores, and from that spore the spores are again germinating to give you a mycelium. So this is the life cycle of reproduction in Phycomycetes. There comes ascomycetes. See, the word asco means sac, and by now we should remember the do you remember the examples of ascomycetes? I will repeat, please repeat with me, and it will get embedded in your head. So there was a boy going to a picnic carrying yeast, edible fungi, morels, truffles. There was this fungi which was giving you this antibiotic called uh, penicillin. So the fungi was penicillium. Then the boy sat for asan, A for aspergillus, C for claviceps, N for neurospora, right? These were the examples. Here in ascomycetes, the hyphae is septate, right? This is filamentous hyphae, and there are walls inside the hyphae, right? This is septate because karyokinesis here has been followed by cytokinesis. The spore can be of both types, sexual and asexual. I am repeating this once again. Sexual spore means the fruiting body was diploid, and the spores are produced by meiosis. And asexual spore means the fruiting body was haploid. So the spores are produced by mitosis. In case of ascomycetes, the sexual spores are called ascospores, and they are produced under a covering, so it is endogenous. In case of asexual spores, the spores we are calling it conidia, and they are exogenous. They are not covered by any covering. No covering is there. And dikaryon is totally visible here. See, dikaryon is not visible. I'm writing it here. Dikaryon is not visible in only Phyco. In the rest of the fungi, like Asco, Basidio, and Asco and Basidio, we get to see a dikaryon, right? So if we look into the life cycle of Ascomycetes, see, is a mycelium, here is again a mycelium, and both the myceliums are septate, right? Septate. So from both the myceliums, we are getting a male gamete and a female gamete. They are fusing. The first step is plasmogamy. Product of plasmogamy is a dikaryon, N+N. Uh, from that dikaryon, the second step, karyogamy, is happening. The two nucleus are getting fused, and we are getting a diploid zygote. Now from the zygote is giving you a mycelium, right? Which is 2n. From that mycelium is growing horizontally, and if you see, there will be again some fruiting bodies. Fruiting bodies are also mycelium only, just they're growing aerially. The As the zygote is diploid, the fruiting body will also be diploid, and this fruiting body in case of ascomycetes we call it ascocarp. Again, carp, whenever you hear this term carp, you get to know that it is a fruiting body, right? From that fruiting body, there are these sac-like things coming out. We call that ascus. That's why, because of this sac-like things, we call this fungi ascomycetes. So this ascus are again deployed, and now see, you see inside this ascus, meiosis is happening. Meiosis is happening, and you are getting the spores, and from the spores, we are again getting a mycelium. And now we are coming to the third phylum, which is Basidiomycetes, right? Basidiomycetes. Do you remember the examples of Basidiomycetes? Basidiomycetes, let's recall the examples of Basidiomycetes. आगरा यूपी में है पर तार से दूर. So agaricus, U से ustilago, P से paxinia, पर के P से puffball, and last ताज के T से toadstool. So this were the examples. In case of Basidiomycetes, the hypha is again septate. So let's draw a hypha, and they are, see, you see that they are septate. There are separations there, walls are there, right? So only the Phycomycetes one was accepted, and the rest all of them are septate. Here a very unique thing happens in this Basidiomycetes, that is somatic fusion of hypha. See, there is no gamete in case of Basidiomycetes, and instead of that, two hypha, suppose this one is a female hypha and this one is a male hypha. These two hypha, they come side by side and they give you a dikaryotic hypha. What is a dikaryotic? This is the result of somatic fusion of hypha, this thing, right? Dikaryotic hypha. So the inside the hypha, the nucleus are not yet fused, they are in dikaryotic form. Do you see? So we what will be the ploidy of this hypha? It will be n plus n. See, not 2n. Note, please note that it is not 2n, it is n plus n, right? And from this dikaryotic hypha, what is we are getting? We are getting a network of hypha. We call that mycelium. Still, the ploidy of mycelium, you see, it is n plus n. That means still no karyogamy. Like karyogamy is delayed here, right? Karyogamy is delayed here. This is a unique feature of Basidiomycetes. From that mycelium, we get to see fruiting bodies emerging. Fruiting body, in case of Basidiomycetes, we will be calling it Basidiocarp. Why Basidiocarp? Because we know carp means fruiting body, and for Basidiomycetes, it will be Basidiocarp. In case of the fruiting body, also the ploidy will be still be n plus n. That means still there is no karyogamy. It is only plasmogamy has happened, and from that fruiting body we get to see this kind of things arising. And now you see that there is only one nucleus I have drawn, there is no two nucleus. Inside the fruiting body also there was two nucleus, right? Because there was no karyogamy happened. But in when I am drawing this branches of basidiocarp, see, I have drawn only a single nucleus. That means karyogamy has happened here, right? And I have marked this as diploid. So delayed karyogamy in Basidiomycetes is a very, very unique feature of Basidiomycetes. And now inside this basidium, these things are called basidium. Basidium. Inside this basidium, diploid basidium, meiosis will happen and it will give you spores. And again, these spores will germinate to give you mycelium. This was all about Basidiomycetes. So now we will give you a summary of whatever we have studied till now. So Phycomycetes, Ascomycetes, Basidiomycetes, and here are the points. In case of hypha, only Phycomycetes, you see, it is aseptate. The rest two are septate. What about asexual spore? How a asexual spore is formed? If the fruiting body must be haploid. So it is giving rise to spores by mitosis, right? So asexual spore in case of Phycomycetes, it is called uh, it is called zygospore. It is called zygospore, and the spore is formed inside a covering, so it is endogenous. In case of Ascomycetes, the asexual spore uh, is exogenous, and it is called conidia. In case of Basidiomycetes, there is no asexual spore. Why? Because in case of Basidiomycetes, there is no asexual reproduction. Also a very important point. There is only sexual reproduction, only sexual, right? Let's come to sexual spore. What about sexual spore in these three three types of fungi. If the fruiting body is diploid, so the spore, which is always haploid, must be produced by meiosis. So let's see what are let's revise what are the three what were the types. So under Phycomycetes, the sexual spore is endogenous, right? In case of Ascomycetes, the sexual spore is endogenous, and we are calling it ascospore. And in case of Basidiomycetes, the sexual spore is exogenous, and we are calling it Basidiospore. What about dikaryon? Dikaryon means it is a structure which is a result of plasmogamy. प्लाज्मोगैमीज का रिजल्ट है ये डाइकैरियन. डाइकैरियन का प्लॉइडी क्या है? N+N, right? So in Phycomycetes, dikaryon is absent. Very important point. Why dikaryon is absent? Because plasmogamy is immediately followed. At the instant at which plasmogamy happens, the same instant karyogamy happens. So there is no chance of producing a dikaryon. In case of Ascomycetes, dikaryon is present. In case of Basidio also, dikaryon is present. The last one is Deuteromycetes. Uh, do we remember the examples? Yes, we do. The trick was cat, C for Coletotrichum, A for Alternaria, T for Trichoderma. These three were the examples. This fungi, the unique point of this fungi is that it is called fungi imperfecti. Why? Because there is no sexual phase. And see, if there is no sexual phase here, so there will be no plasmogamy. If there is no plasmogamy, there will be no dikaryon. If there is no dikaryon, there will be no karyogamy. No zygote, no meiosis, right? Because there is no sexual phase. That's why it is called fungi imperfecti. We have seen that this no dikaryon, this point is also same for Phycomycetes. But then how is it different from Phycomycetes? Because Phycomycetes was accepted. This hypha was continuous, sinusoidal, accepted. But in case of Deuteromycetes, we see that the hypha is septate. That is, there are partitions all over there, right? Okay. So there is only this asexual spore which is called conidia, right? And Deuteromycetes is thus a temporary phyla. Suppose you are a researcher of this researcher doing your research in fungi, and you get to discover a totally new fungi, and you see that here these points, the fungi is septate, but it is septate means that it cannot be not be Phycomycetes, but the fungi is not showing sexual, still not showing any sexual phase, right? It might show sexual phase in future, but the moment you discovered it, it is not showing any sexual phase, and it is showing only asexual phase for now, and those asexual spores are conidia, right? So you will place it under Deuteromycetes. Why not under Ascomycetes? Because it is, for the time being, it is not showing a sexual phase. But हमको नहीं पता ये future में जाके sexual phase दिखा भी सकता है, right? तब तब हम उसको एस्को में रख देंगे. अभी के लिए सिर्फ asexual phase ही दिखा रहा है. So we are placing it under Deuteromycetes. We will be revising the whole whatever we studied for now, that Phyco, Asco, Basidio. This is a summary kind of a slide. So habitat, what is the habitat? Aquatic for Phyco, coprophilous. What do we mean by coprophilous? Philous means it feeds on, and copro means dung. It feeds on like dung, cow dung or something like that. It feeds on there. Basidiomycetes habitat, log, tree, soil. Dikaryon, very important point. Dikaryon is not present in case of Phyco, but it is present for Asco and Basidio. Structure, it is in case of Phyco, the hypha is aseptate, okay, but in case of Asco and Basidio, there are partitions, septate, right? Nutrition, all of them are saprophytic. If we come to the reproduction, which is very important point, vegetative reproduction is seen in Phyco, that is fragmentation, also for Asco and also for Basidio. Asexual reproduction, Asexual is seen only in Phyco and in Basidio. In Basidiomycetes, there is no asexual reproduction. In Phycomycetes, the spore is called sporangiospore. The spore is formed inside a covering, thus that's why the spore is endogenous. In case of Ascomycetes, the asexual spore is called conidia, and uh, it develops without a covering, that's why it is called exogenous. What about the sexual spore? Sexual spore in case of Phycomycetes, we call that zygospore, and it is endogenous, right? In case of Ascomycetes, the asexual spore is called ascospore. It is again endogenous. And in case of Basidiomycetes, the sexual spore, it is called basidiospore. It is exogenous. There is a trick, small trick. If any spore starts with A or Z, that is ascospore, like zygospore, it must be endogenous. Right? This is a small trick. If any spore, be it asexual spore, be it sexual spore, if the spore starts with these two letters as A or Z, it must be endogenous. Apart from that, if the spore starts with any other letter, that is suppose basidiospore starts with B, it must be exogenous. A small trick, you can remember it. So that was all about the kingdom fungi. I revised it. It was a very short video, and yeah, hope you like it. And if you liked it, please hit the like button, and you can motivate me by commenting in the comment box. Thank you.